bachelor in biotech engineering, and this was my final year project work, and it is about the study of effect of probiotics in developing resistance against WSSV in shrimps. You must all have heard about shrimp farming. It's so common in southern parts of India and everywhere, like countries like Japan, Australia, UK, not in UK of course. And it has been, this sector has been popularized in India from early 1970s. So what is shrimp farming? It is the growing of shrimps for human consumption. That is what is shrimp farming. And it's a major economic and food production sector. And India is the 5th largest exporter of shrimps in the world. And this data, that is 357,505 metric tons of shrimps have been exported in the year 2014-15 alone. So it's exported mainly because of this demand in the foreign countries, especially in Europe, and also because it's an important source for human protein, which is available for human consumption. But as shrimp farming has been popularized, as the number of shrimp farmers increase in our country, the challenges faced by them also increase. The major challenges includes the diseases caused by viruses, bacteria, and fungus. White spot syndrome is one of the major challenges faced by shrimp farmers and is causing lethal reduction in the number of shrimps in our country. This is a disease which is caused by a virus called as white spot syndrome virus. It's a double-stranded DNA virus and it belongs to the family Pneumaviridae. There are 2 methods of transmission of this virus. This virus can be transmitted in farms through, that is called as vertical transmission, that is by oral ingestion of infected shrimps. That means the shrimps eating the dead body of infected shrimps. That's through oral transmission, or it can also be transmitted through infected water. And in shrimp hatcheries, vertical transmission takes place, that is from an infected mother to its offsprings. So there are 2 modes of transmission of this disease. The traditional methods now available for controlling or prevention of disease is very limited. We just select the post larvae which are virus free, so that the next coming population or the next generation won't be affected by this disease. And also we treat the water and we treat the wastewater so that this virus can be eliminated, and by this way we can reduce the concentration of virus in the water. Effluent management is also a method which can be followed, in which biological filters, that is fishes like tilapias and milkfish, can be used for reducing the effluents in the water, present in the water. The use of bacteriophage has also been come up recently. The use of bacteriophage is very useful because bacteriophages are organisms which consumes infection-causing bacteria and thereby helps to reduce the occurrence of diseases in shrimps. Also, there are many chemicals and many other compounds which can be used so as to reduce the concentration of diseases or occurrence of diseases in shrimps. But all these have many problems. The main problems include is that when you use antibiotics or chemicals so as to eliminate the occurrence of this disease, this causes problems for other aquatic flora and fauna. So what came to our mind is using probiotics. If we use probiotics to improve the resistance of shrimps against this disease, then the other aquatic flora and fauna won't be affected. So that's why we came to this idea by using probiotics for improving the resistance of shrimps against this disease. As you may be knowing, probiotics are live organisms which gives health benefits to the host. It is very common in the case of warm-blooded animals, it's very effective. But in the case of shrimps, we don't know whether it's effective or not because the immune system of shrimps is very weak and it has not been studied much. So we tried to study whether probiotics have any effect in developing the resistance of shrimps against this disease. For this, we used freshwater shrimps of almost 4 to 6 centimeters. We collected these shrimps from shrimp farms near Velaini in Trivandrum, and these were brought and acclimatized to the experimental conditions. And we also prepared a medicated diet. The medicated diet means it's the diet in which basal diet is dried coconut flakes, but in the medicated diet we incorporated probiotics like yeast, yogurt, and curd into the feed. So these were the shrimps which we used for the experiment. And the experimental design was that we took plastic containers which contains almost 3 liters of water, and we took almost 2 shrimps in each of these containers, and we separated these containers into 4 groups. In the first, we added basal diet incorporated with yeast. In the second one, we added basal feed incorporated with curd, and in third, the basal feed incorporated with yogurt. The fourth group was set as control, in which only the basal diet was provided for the shrimps as feed. This experimental feed was given to these shrimps for almost 10 days. After that, and during this process, almost 30 to 40% of water was changed all day so that the shrimps gets more oxygen, and continuous aeration was given through oxygen pumps, aquarium air pumps. The next step of the project was to challenge these shrimps with white spot syndrome virus. This has been done by giving the shrimps infected dead bodies. That means infected prawn flesh were given to these shrimps to feed so that these gets infected. And the post-challenge survival rate was recorded. This was the observation which we obtained. Here we have the container numbers that are, I told you that 3 containers were used for all the 4 sets. This was a controlled yeast, that means the basal feed in which yeast was incorporated. This was the one in which the basal feed curd was incorporated, and the last one, the basal feed in which the yogurt was incorporated. And this 2 shrimps were taken, and these were the number of days which the shrimps survived after challenging with the virus. In the control, shrimp 1 survived for 7 days and shrimp 2 survived for 8 days. And in the container 2, 5, 7 respectively, 9 and 6 respectively in the third container, which gave an average of 7. Like that, for yeast we got an average of 6.5, and the case of curd we got an average of 21.5, and the case of yogurt we got an average of 10 days. From this result, it was so clear that the basal feed in which curd was incorporated give the maximum number of days. That means the shrimp survived for the maximum. That means almost 21.5 days in the containers in which curd was incorporated as supplement. So the average days, number of days in which the shrimp dies is around 6 to 10 days. But when curd was incorporated as supplement in the basal feed, the shrimp survived for 21.5 days. So it is so clear that when curd is incorporated as supplement, it will increase the lifespan and it will increase the resistance against WSSV in shrimps. So this was our conclusion, and I think that this will be very helpful for the poor shrimp farmers in our country, and it will reduce their economic losses and thereby increase their profits. Thank you.